It's time for another warm-up review. Let's get going. Which muscles and nerves are derived from the second branchial arch? So the mnemonic for the second arch structures is the letter S itself. So you have cranial nerve 7, the facial nerve, and that innervates the muscles of the facial expression. Then the S muscles include the stapedius and the stylohyoid. Next, what is the antidote for each of the following substances? So first we have opioids, so either naloxone or naltrexone. Antidote for benzodiazepines, that's flumazenil. For heparin, the reversal agent is protamine. Warfarin interferes with the liver's ability to make vitamin K, dependent coagulation factors, so the reversal agent is just going to be vitamin K. But since that actually takes several days to really improve coagulation, for immediate reversal of warfarin, we give a prothrombin complex concentrates to replace those coagulation factors that the patient is deficient in. And the last one here is theophylline. You're going to use beta blockers for that. Next, which ECG finding is associated with hyperkalemia? So remember that T waves mirror the potassium level. So with high potassium, you get tall peaked T waves, and with low potassium, you get kind of flattened T waves, maybe also a U wave. All right, that's it for the review. Let's get to that video.
In this video, we're going to discuss the details of antigen presentation. In order for your body system to mount an immune response to anything, the immune system has to recognize it as a threat. For example, a phagocyte, like a dendritic cell, engulfs some pathogen, like bacteria or a virus or whatever, and then digests it. And then the dendritic cell puts a little piece of that pathogen on the surface of the dendritic cell, where the helper T cell can see it and bind to it. So dendritic cells are antigen-presenting cells that are presenting a foreign antigen to a helper T cell. That whole point is to activate the helper T cells, to tell the immune system that there's something, a threat, that they need to start attacking. We'll review the different kinds of T cells and the activation of T cells in another video, but for right now, I just want to go over a couple of really basic points about T cells that apply to the antigen presentation. A T cell precursor begins life in the bone marrow, and then they have to travel to the thymus, where selection takes place, and the cells that are selected to mature into T cells, and the rest that aren't selected, are destroyed. We're going to talk about that later. Remember, they're called T cells because they mature in the thymus, and T is for thymus. Now, a T cell is a type of lymphocyte, but you can't tell a T lymphocyte from a B lymphocyte just by looking at it. So how can you tell them apart? You tell them apart by the different surface markers and receptors on the T cell, and these surface markers are worth knowing for step 1. Every T cell has a very specific T cell receptor, or TCR, that's capable of binding to a very specific antigen. Remember, T cells are a part of the adaptive immune system, which is very specific. So, T cells have a T cell receptor that can bind to a specific antigen. And actually, there are hundreds, maybe even thousands of T cell receptors on the surface of a single T cell, and they're all identical. This T cell might have thousands of receptors for an antigen that's found on the chickenpox, the varicella virus, and this other T cell has thousands of receptors for the measles virus antigen, and so on. Very closely associated with the T cell receptor is a surface marker called CD3. It actually helps the T cell receptor transmit a signal to the T cell when it binds to that specific antigen. So I want you to think about CD3 as a marker for all T cells. Now, just in case you thought this was getting a little too simple, there are different types of T cells, which can also be differentiated by their surface markers. We mentioned helper T cells. Helper T cells have a CD4 surface marker. They're sometimes abbreviated TH, so sometimes they get called T helper cells. Helper T cells or T helper cells, same thing. The other big type of T cells are cytotoxic T cells, which is abbreviated TC. Cytotoxic T cells are identified by a CD8 surface marker. There are also memory T cells and regulatory T cells, which are supposed to be called suppressor T cells, and a few other types that you don't need to worry about for step 1. Thank goodness. Again, the term lymphocyte refers to both T cells and B cells, but around 80% of the lymphocytes in circulation are actually T cells. So when someone talks about lymphocytic infiltration, for the most part, we're talking about T cells. Let's talk about how helper T cells get flagged down by an antigen-presenting cell. Basically, the T cell receptor on a helper T cell will bind to a specific antigen only when that antigen is presented by an antigen-presenting cell. This is because the CD4 marker on a helper T cell is actually very closely associated with that T cell receptor. And the CD4 marker actually links up with a protein complex on the surface of the antigen-presenting cell. This is called the major histocompatibility complex, or MHC, specifically MHC class II. The way this works is after the antigen-presenting cell phagocytoses that pathogen and digests it, it puts a piece of the antigen on the surface attached to the MHC2 molecule. Then, that antigen-presenting cell can start presenting this antigen to every T cell it meets, looking for one with matching T cell receptors. When it finds one, the CD4 on the T cell has to bind the MHC2 on the antigen-presenting cell. And when that happens, the CD4 and CD3 can start their signaling cascade that ends up activating the helper T cell. And we'll talk about that activated helper T cell in another video. But for now, just remember that CD4 positive helper T cells bind to MHC class II. In the video, when we talk about T cells, we're also going to look at cytotoxic cells. Now, if cyto means cell, what do you think cytotoxic T cell does? It kills the cells. Specifically, cytotoxic T cells are supposed to kill virus-infected cells, or cancer cells. Well, that's all well and good, but how can the cytotoxic T cells recognize the virus-infected cells or cancer cells? How can they tell the difference between a healthy cell and a diseased cell? Well, the answer is the major histocompatibility complex again, but this time it's MHC class I. MHC class I is expressed by basically every nucleated cell in your body. So not the red blood cells, but pretty much every other cell in your body has an MHC class I molecule on their surface. And attached to those MHC class I proteins are peptide antigens that come from inside the cell. Your antigens are slightly different from mine, so those unique antigens get attached to something called the peptide binding groove on MHC class I molecules on the surface of all of your cells. It's kind of like a way for your cells to tell the cytotoxic cells, hey, leave me alone, I'm on your team, I'm one of the good guys. But when a cell is infected by a virus, it starts putting viral peptide antigens in the peptide binding groove of the MHC1 molecules. And that tells the cytotoxic cells, psst, virus, I've got a virus. And the cytotoxic cells are going to recognize that viral antigen and kill the cell. Because remember, a virus-infected cell is just a factory for more viruses, and the only way to deal with it is to kill the infected cell. It's the same kind of thing you do with a cancer cell. Cancer cells start expressing weird cancer antigens in their MHC class I molecules, and that tells the cytotoxic T cells something's up, so they can come attack and kill that cancer cell. So think back. If CD4 was the surface marker for helper T cells, what did we say the marker was for cytotoxic T cells? That was CD8. So just like the CD4 binds MHC class II on antigen-presenting cells, CD8 binds MHC class I on virus-infected cells, or cancer cells, and then the cytotoxic T cells kills those cells. It's pretty easy to keep this straight in your head. You just multiply the CD number by the MHC number, and you get 8. CD4 times MHC2 equals 8, and CD8 and MHC1 also equals 8. How convenient is that? Now let's dig into some genetics of the major histocompatibility complex. Like all proteins, MHCs are derived from very specific genes. The genes that code for MHCs are called HLA genes, human leukocyte antigen genes. And just like your MHC1 molecules are unique to you and different from my MHC1 molecules, your HLA genes are different from mine. Again, MHC1 molecules are found on virtually every nucleated cell in your body, and they are coded by 3 genes: HLA-A, HLA-B, and HLA-C. And then HLA-D codes for MHC2. But we have to break them down into 3 subgroups: Remember, MHC2 molecules are only found on antigen-presenting cells, and the genes that code for MHC2 proteins are HLA-DR, HLA-DP, and HLA-DQ. Now, all of these HLA subtypes can be further classified into specific loci and specific alleles. You might divide HLA-DR into HLA-DR1 or DR2 or DR3, and so on. Or, for HLA-B, you can have HLA-B1, HLA-B2, and so on. That's where HLA-B27 comes from, which should sound somewhat familiar to you. The importance of this is that certain alleles and loci are associated with certain diseases. And not too surprisingly, these are generally autoimmune diseases, where the patient's own immune system is attacking from within. And not all of you will have a question associated with HLA and a certain disease, but a few of you will. And the ones that are important to know are HLA-B27, HLA-DR3, and DR4. As we discussed in rheumatology, HLA-B27 is associated with seronegative spondyloarthropathies, and the mnemonic for these is PAIR. The P is for psoriatic arthritis, the A is for ankylosing spondylitis, and the I is for the arthritis you can see with inflammatory bowel disease. And R is for reactive arthritis, or the older name was Reiter syndrome. And then HLA-DR3 and DR4 are associated with type 1 diabetes, which is caused by autoimmune destruction of the pancreatic beta cells. Cells that express MHC2 on their surface are antigen-presenting cells. And there are a handful of cells that can act as antigen-presenting cells, like macrophages, sometimes certain B cells, but by far the most important type of antigen-presenting cells are the dendritic cells. Let's look at number 4 in your study guide. They're called dendritic cells because they're covered with these membranous extensions that look like dendrites on a neuron. And they've got their feelers out, trying to capture any antigen that may come along. Dendritic cells display both class 1 and class 2 MHC molecules. Class 1 because all nucleated cells display MHC1, and class 2 because that's how they present the antigen to the helper T cells. They also have a surface protein called B7, which can bind to the specific proteins on the T cell and either increase or decrease the signal that is activated when the T cell receptor binds to the antigen that is being presented. And B7 is sometimes called a co-stimulatory molecule. It won't activate the T cell by itself, but it can influence the activation of the T cell when other signals are around. Dendritic cells also have a CD40 surface marker, which allows the T cells to activate the antigen-presenting cells. So everybody is stimulating everybody else. We're going to see CD40 and B7 again in the next video. This is just a brief introduction. But CD40 is found on the dendritic cells, but it's also found on other antigen-presenting cells, like macrophages and B cells. So what do dendritic cells do? They're designed to capture antigen. They capture antigen at one location and then present it to T cells in another location. Once a dendritic cell captures an antigen, it changes its shape. It's no longer a cell with those long dendritic arms. It changes the shape so that it can migrate to the lymph node and say, hey, look what I found! I found some antigen. Out in the periphery, dendritic cells serve as peripheral sentinels. A sentinel is like a guard or a watchman or a sentry. This is sometimes called the immature state, or the sentinel state. And in that sentinel state, that antigen capturing state, they can capture antigens by 3 different mechanisms. By phagocytosis, by receptor-mediated endocytosis, where the dendritic cell's surface receptors may recognize an antigen and then endocytose that antigen using clathrin-coated pits. And the third mechanism is penocytosis, which is like endocytosis of extracellular fluid and whatever small particles are floating within it. Kind of like cellular drinking of fluid, dendritic cells can replace their entire cell volume within an hour through penocytosis. So it's kind of like a sieve. It's just taking a bunch of stuff in and looking for any antigen it can find within that fluid. So the cell starts in this immature sentinel state, this antigen capturing state. Then, once it's captured an antigen, it makes a transition to an antigen-presenting state, which allows it to travel into the circulation. And in this state, the dendritic cell has maximal capacity to present antigen to the helper T cells because it's increased expression of class II MHC and increased expression of those co-stimulatory molecules. So they find an antigen and then they switch this cell that presents very well to the T helper cells. There are also different types of dendritic cells. One you may have heard of is the Langerhans cell. A Langerhans cell is a dendritic cell that's found in the epidermal layers of the skin. Sometimes it's referred to as a cutaneous associated lymphoid tissue, or CALT. So the Langerhans cell is a type of dendritic cell that hangs out in the epidermis, and again, once it finds an antigen, it's going to change shape and migrate to the lymph nodes. And then in the interstitial spaces of your organs, except the brain, you have interstitial dendritic cells. Let's briefly discuss a pathology that can arise in the Langerhans cell, which is called Langerhans cell histiocytosis. This is a disease where there is an excessive proliferation of Langerhans cell, these dendritic cells in the epidermis. And the problem is these cells are no good at antigen presentation, so they're not able to do their job. These cells express an S100 tumor marker and also a surface marker called CD1A. And the way you would probably see this on your test is that in electron microscopy, these cells are going to have a Burbeck granule, which look like little tiny tennis rackets within the cell. That's it for antigen presentation, so pause the video and try to answer the end of session quiz. First question. What molecules are expressed on the surface of dendritic cells? We mentioned MHC class I, MHC class II, a B7 protein, and CD40. Next, what are 3 cell types that are known for presenting antigens to CD4 T cells? These are dendritic cells, macrophages, and B cells. Next, what characteristic finding would you see on electron microscopy of a dendritic cell in a patient with Langerhans cell histiocytosis? That's a Burbeck granule. Last one. Which MHC class is recognized by helper T cells and by cytotoxic T cells? Helper T cells recognize MHC class II, and cytotoxic T cells recognize MHC class I. That's it for the session quiz.